What
Okay,
is a
so
tetrad?
sorry, let's go back
So
to
if we
draw
look
each
over
phase of
here, oops, that's not
meiosis.
the one. If we look
That
up
just
here
didn't
to
go
the
down
pictures,
far
that's a
enough. Here we
tetrad.
go.
Tetra
All
means
right, so
what?
here
It means
is,
four.
you
So
want
this
to
is a
be able to
tetrad.
recognize the difference between
And
meiosis
what
and
it is a
mitosis,
four-part
and you want to
structure that's going
know
to form
everything that's going
during
on in there.
prophase
So
of meiosis.
in
And it
meiosis,
consists
in
of
this
two
process, you
homologous
can see we start
chromosomes. So
off
here's
with
one homologous
diploid,
chromosome. Here's
2N,
the other homologous
and
chromosome.
we go through
And each
replication, and
homologous,
so both of these are the same at that point. And so
each
after
homologous
meiosis
chromosome
1, we
is
have
composed of
haploid
two
cells
sister
versus
chromatids. That's just a
mitosis, we
regular
still have diploid, 46,
definition of
46.
a
So we go
chromosome of
diploid
two sister
to
chromatids.
haploid,
But
and
now
then
with
we
a
end up
tetrad,
with
you have
4
two sets of
haploid
those.
cells. So they all
All
have
right.
23 here.
Ding. Let's go back here.
And
all right. What's the
so
difference
there's our
between
picture of the difference between meiosis and
translocations,
mitosis. Of
and
course, mitosis
deletions?
occurs in the somatic
From
cells,
our
also called the autosomes,
activity
and
yesterday with
meiosis
disordered
occurs
detectives,
in
clearly we were able to
the
see that an
sex
inversion
cells
is
in the
part
X
of a
and Y
chromosome,
chromosome.
breaks
All right.
away, and reattaches to the same
Let's see, where did
chromosome.
we end
That's
up
the key.
with?
It's on the same chromosome.
Oh, we were talking
Where
about
when you
law
have
of
a
independent assortment. So we're
translocation,
at
you have
dominance
part
and
of the
recessive from
chromosome,
looking at a pedigree.
that goes from
Well, there's a couple
part of
things.
one
We could look
chromosome
here
then goes
first
over to
at
another
these
chromosome.
pedigrees, looking at
All right, in a
autosomal
translocation.
and
And
dominant
inversions
and
on the same
autosomal
one, so you have the
recessive.
movement of the
But
genes
really,
on that
I like
chromosome.
this one better.
Translocation, you have a segment of those genes
This is
travels
looking
to
at
another.
pedigrees
So with
in
a
general. So
translocation, you
here
can see one
is
that's
whether it's
larger and
autosomal
one
or
that's
X-linked,
smaller
and we
in
know
there.
if most of the males
And
in the
then with a
pedigree are
deletion,
affected, then
it
the
is
disorder
just
is
as
going
it
to be
says,
X-linked.
part of the chromosome.
If it's a
or part
50-50
of that
ratio, then we
sequence
know it's going to be
of
autosomal.
DNA
So just looking
is,
at it, do you have more
is
of one
deleted.
sex
So
that
let me
has
show you
it?
here a picture, possibly.
I'm not sure
If I
why
can, oops, I got to get out of this.
this is
Here we go.
not
All
doing
right.
that. It
There.
froze, of course.
All right, so if we go down
See
here,
if I can
you can see
get it to, there we go. All right.
this is a
Now,
deletion.
determining whether or not
the
it's
deletion,
dominant or
and then
recessive, if the
this is
disorder is
after the
dominant,
deletion,
one of the parents
because
must have the
the,
disorder. If it's
deletion,
recessive,
you can see how
neither
this is
parent has
short.
to have the disorder, but it can
then
be heterozygous.
here,
So
with an
you can look
inversion,
here. One of the
this
parents
is
has it.
just a paracentric one. It's just an
and
example. But this region
in each one. Now
is
let's
now
look
up here.
here.
So you can
This
see the
is
banding would look
all right.
different.
So here are some
and
basic
they're
rules of
just
inheritance.
inverted.
If it's
But it's still
autosomal
on that
recessive,
same chromosome.
it's going to
This
appear in
isn't going
both
to here, and
sexes
this
with
isn't going
equal
to here. This
frequency,
is
and the
chromosome,
trait
and
tends
then
to skip
on the
generations.
other one, it's
So the
inverted.
rest are
right.
important, but those are the keys. Because
and
look
then
at
over
autosomal
here,
dominant.
we have
Again,
our,
appears in both sexes with equal
translocation.
frequency,
So we
but
have this information
it does
going
not
over
skip
to here.
generations.
So we see this
All right.
area here.
Now, if it's
going on over,
X-linked,
and you can see
both males and females are
switch
affected,
there.
but often more females
All
than
right.
males are affected.
Describe
And it does not
crossing
skip
over and
generations.
its importance. So
X-linked
crossing
recessive,
over,
more
if we look at this
males than
picture right
females are
here,
affected. And
there we go.
affected
Okay, so
sons
here's
are usually
crossing
born
over.
to unaffected
And
mothers.
it's going to
Okay,
occur
so
in
that
prophase
should
1.
help. Let
And
me
we
see
have
if they
two
have. Okay, so
chromosomes
here
cross
is a
over and exchange their genetic information.
an
So here's the
X-linked
tetrad,
recessive.
and you can see where
only
they cross over
males
here.
are
So we have
affected.
blue crossing over red.
You can have,
those aren't the real
and then
colors
here's
of the
X-linked
chromosomes,
dominant.
But we do that so that
So
we can
father
demonstrate
passes
what's
it on
happening.
to only.
All right, and so and then you have
on to
in
daughters.
metaphase 1,
All right.
you start to have
Here's
these pulling
autosomal
apart
recessive.
at the kinetochore,
Affected parent.
at the
Affected individual
centromere there.
comes from
and then you have,
unaffected parent. So
and you can
it's
see
skipping
when,
that
when
generation.
these are pulled apart, now
And then
you have
autosomal
red tips
recessive,
on the blue chromosome
you're going to
and
have
vice versa. So this genetic material
expressed
from this
equally
one is now over
in
here.
both sexes,
So
again, because it's
not only
autosomal.
are you getting chromosomes from mom and dad, are you getting
autosomal
information from
dominant.
mom and dad? But now
It's in every
even within
generation.
mom and dad now
And
we're
because it's
mixing
autosomal,
everything up.
it's equal
And
in males
ultimately,
and females.
we end up with
Autosomal
the,
dominant, again,
4
appears
haploid,
equally in males and females.
cells, and these two
okay.
have
so that
more
should
genetic
help looking
variety
at
to them.
the difference
right.
there. What else do we
Okay.
have? Autosomal or
Look at my
sex link. So
timer
we did
here.
all of it.
How much time? Okay.
We did 14,
Now
15.
we're moving on
Be able
here
to
to number
determine a
6.
genotype from looking at a person's
what
affected or
gametes are
non-affected
produced
condition on a pedigree.
from
We practiced these. I'm telling you guys, the best way to
the other
practice
thing-
it is do
sorry,
more.
going back
Just
to
find some on
number
the
5,
internet,
it's important.
practice them, or use the ones that we
genetic
had.
variety
and
the variation. Remember,
and cover up
we've
your
talked about this
answers and
several times
practice
where we've talked about
figuring
why
out
that
what
variety
those
is so important,
genotypes are
especially,
going
when
to
we
be,
talked about asexual versus
whether or
sexual
not they're affected
reproduction.
or not.
We need the variety because if one
describe-
disease comes through and it's
Did I
going to
put
wipe
that on
out
there
one,
twice?
it would wipe out
I sure
everyone.
did. So number 4 and
as far
number
as survivability,
17
the more variety there
are
is,
the same.
the higher the survivability
Number
rate.
18, determine
All right,
possible
let's go on to 6
offspring
now. What gametes are
genotypes
produced from
related
a
to
heterozygous
blood
genotype,
type.
homozygous
Again, guys,
dominant
this is
genotype,
working that
and
same
a
problem
homozygous recessive
like we did in
genotype.
class, in the packet.
Well,
So,
ultimately,
can you
if
work
we
blood
have,
typing problems? Make sure you
a
practice
heterozygous,
those.
that's
You're
capital
looking
letter
at
and
your
small, dominant and a
A,
recessive allele.
B, O, and AD blood
and
types.
You want to look at
why
what
are
is produced.
X
So if
and
we
Y
have
chromosomes
heterozygous
considered
crossing,
sex
we're gonna get,
chromosomes? Well, they're considered
from heterozygous,
sex
one
chromosomes
allele
because
is
the
going to be
genes on those chromosomes interact with other
capital
genes
letter,
scattered
a dominant
throughout the
allele. The other
genome,
one is a
and they
lowercase,
determine the
that's gonna be a
individual sex.
lowercase
So they're
allele.
still
So
going to
heterozygous,
interact with the
we're
other
gonna
one. They
end up
still have
with
there's
big
still an
P,
impact on the
little
other genes,
so
but
one
it's going to
alleles
determine
capital, one
sex
alleles
of the
lowercase.
individual. Whoa,
With
where'd it go?
homozygous dominant,
Going down here.
both capital
All right.
letters.
In
And
terms
with
of
homozygous
genetics,
recessive, obviously,
what are the
one
advantages
allele,
and
that's a little p, we'll
disadvantages
make
of
that a
sexual
little p from
reproduction?
there.
Well,
One little p and another little p.
first of
right?
all, sexual reproduction, the advantage is
Okay.
you have a
Away we go.
mixing of the chromosomes. You have the mixing of the genetic material.
7,
And
red-green
so,
color blindness.
you have,
If
you're
we're
getting some from mom,
describing
some from dad.
this,
And so you have the variety. Remember we talked about how
we are
important the
looking
variety is
at,
for
we want
survivability.
to look at
So that's
and
an
describe
advantage. A
everything related
disadvantage
to
is
red-green
it
color
takes
blindness.
much
Why
longer
do
to
we have
find a
it?
mate. Especially like in
and
the
be able to
courtship
understand the planets
stage,
we're
if there's something
responsible
like
for it.
that
So when
involved.
you're looking at red-green color blindness,
And so you have to
first of all,
physically
you're
find a
probably gonna
mate
notice that
and
it's gonna be a
then
sex-linked
woo
trait.
them and then
and
hopefully
you're gonna follow,
make offspring from that relationship.
just
Where
make
with
sure that you can work
asexual
the planets where
reproduction,
related to red-green
there's
color
no finding a
blindness.
mate. You just
All
instantly
right,
can
number
make it. So it's a
8,
lot
the
faster
phases of meiosis.
form of reproduction. However, there's
All right.
no exchange of genetic material. I'm sorry.
I-
There's no
had it in
variety.
Where did I
And
put
with
it?
that, it becomes very dangerous because one
I
thing
don't
comes
know
along
where I
that's
put
going to
it.
wipe it
Okay.
out. And it will wipe
Let's
everyone
skip
out.
that. I'll come
all
back
at
and I'll
once.
find it and I'll put it in the
All right,
next video.
21. What would a
right,
chromosome look
sorry about
like
that.
in an egg or a
La,
sperm?
la,
Let me go
la,
to
la, la,
those
la.
pictures.
Where's
Where
the
we at
next
here?
question?
Pictures. All right. So chromosome would look like in an egg or a
Where
sperm.
are
All right. So
alleles
here's
found?
an
Where are they
egg.
found?
Here's a sperm.
They're
That's what
found
they
on
look like.
homologous
They're
chromosomes
haploid.
Is where
Okay. Individuals.
alleles
The
are found.
zygote is the fertilized egg. So after the genetic material from the sperm has gone into the egg,
Number
this is what
11.
the zygote looks like.
What is the purpose of meiosis?
Then last but not
Well,
least,
we
how
have to
could
have a
members of a
reduction in
different
the number
pair
of
of
chromosomes.
homologous
We can't have,
chromosomes
in the chromosome number. We can't have 46.
differ
because
from
if we had
each other?
46 sperm,
Well, they're going to
46
differ in
chromosomes
that
in the
sequence
sperm,
of
46
the
in
precise
the egg,
sequence
we
of the
would end
DNA
up
that are
with
within
92,
each chromosome.
and that
So
would not be a viable human.
they're going to have
And
some
so it would not survive.
variability
And so,
there.
We have to have
All right, I think that
this
covers it all.
reduction in the number of
Test is
chromosomes.
Tuesday.
And we also have the
Test is
variability.
Tuesday. And bring
It-
your study
we end up having a
document.
set of
That's your ticket
cells
to
that
take the
have a very
test.
unique set of genetic
Missed you
material.
guys a little bit. I mean, like I really, I'm not
What
really
is a
thinking of you a
homologous
lot,
chromosome?
most likely at this point,
Homologous
because
chromosome is
Rome
a set
is amazing.
Of one
Arrivederci,
maternal
everyone.
chromosome
See you
and
Tuesday.
one paternal chromosome.